Auzubillahi Bismillahirrahmanirrahim. I hope you had a very good cup of tea, and now it's time again to be here. And I think once we join such meetings, the most precious things we can get is the latest studies we learn from there. Latest, because most of the stuff we hear it is not available in literature. Uh, that's what I have seen in most of the researches, that when we listen those researchers in such conferences, they were novel studies. So this is one of the novel study. Most of the content I'm going to show you in this study is not published yet. So we have come up with some interesting data. So I hope this 30-minute session, I would just appreciate if you can be connected. And then once you are connected, then it's my responsibility to keep you along. So the topic today we are discussing is basically Salmonella Gallinarum. We have been working on its vaccine for last 8 years, and this is our outcome of last 8 years work. Those of you who are interested in working in Salmonella, they already know its basic thing, that Salmonella can be divided into 3 categories. The one we call the general Salmonella, like Salmonella typhimurium. If you look at these, they are less pathogenic, more generalized. Then pathogenicity increased and specificity increases. So then we have a group of Salmonella like Dublin and Chlorisues, which are mostly host adapted. The very much host restricted are those which cause typhoid in their specific species, like Salmonella typhi in human, Gallinarum and Florum in case of poultry. So they are limited to their hosts. They do not move around in various hosts, but they are very dangerous. So what they do, that generally they change their genome from general genome to specific genome. So that's what we have seen. So here in this study, we are specifying one of the organisms, that is Salmonella Gallinarum, which is host specific and fowl typhoid organism. If you look the global status of this organism, it has got 2 biovars, Salmonella florum and Salmonella Gallinarum, and you can see the distribution of this disease in the globe. If you look into the various patterns of this disease over last 40 years till 2022, this Chinese paper says that mostly it was the Salmonella florum which was the prevalent organism, but later it is mainly being covered by Salmonella Gallinarum. So we are seeing more Salmonella Gallinarum in those countries only where the Salmonella is present. So we are seeing more Gallinarum as compared to florum for some reasons which we are not going to discuss over here. So keeping this in mind, that Salmonella Gallinarum is the pathogen of future as compared to florum, so we have designed that how we can add value to the scientific community, especially the poultry industry, that this Salmonella can be eradicated or controlled with the help of the vaccines. So there are different ways to control Salmonella. Those who have worked on farms in poultry, they know that how important it is to work in such a way that you can control Salmonella. Again, when we say the control of Salmonella Gallinarum, that has a different reason for the control in poultry side. And when we talk of pathogens like enteritis and typhimurium, that's a different reason. But here we are seeing that how Gallinarum can be controlled. So the vaccines we are talking about can be many types, but live attenuated vaccine is one of the area where I really like, because I have been in debate. The subunit vaccines and kill vaccines, they have their own benefits. But in poultry, I believe that the whole bacterial organism provides a diverse type of immunity which keep you more protected. So that's what I believe, that if you have a local antigen, local bacteria, and you are capable of changing it into the vaccine, that is the best way to vaccinate your local birds or animals. That was the idea behind this study. So if you look into the current market of Salmonella, mostly the Salmonella Gallinarum, one vaccine is usually used globally, and that is the strain called SG9R. So this is Novel's product, but many other companies make product out of SG9R. SG9R is the strain, which is a live Salmonella Gallinarum vaccine strain available all over the world. There is another strain I have heard, which has been patented by Petro. I have discussed over here. But other than that, there is no Salmonella Gallinarum vaccine, and mostly Salmonella typhimurium and Tritidus vaccines are available, which are gene-deleted vaccines. So our idea was to create some potent gene deletions, which should be the novel gene deletions, so we can get a patent out of it, and we can make it a vaccine from our local pathogen. So there were three objectives we have covered in this study. In the first study, then we'll move to the second one. So we plan to knock out two important genes from Salmonella to make it a double mutant, and then we try to study their pathogenicity, whether after deleting those genes, the bacteria was capable of producing any disease or not. If it is not, it's safe, it can be used as a vaccine. And third, if it is safe, whether it really immunizes your bird. So these were the three objectives we studied. So the Salmonella we picked for this work was one of the isolate from Pakistan. And this was the second isolate we were able to sequence. And there are only mainly two sequences submitted from Pakistan. They both were from my lab. And this is the latest sequence submitted from Salmonella Gallinarum. So we used this organism as a basic organism towards building the genome deleted. So this is the most critical part when you work on gene deletion, that you have to decide that what genes to be deleted to make it a vaccine. That was the question. So if you know the physiology and pathology of the way organism cause disease, so one of the major pathogenicity factor is type 3 secretion system in Salmonella. Those who work in microbiology, they know this type 3 secretion system. So what we did that we picked one of the gene SSAU, which is responsible. If you look at this picture, you can see, if I had got a pointer, I can show you, but otherwise there is a area in the center where this area, this is transcribed by a gene called SSAU. If you knock out this gene, this whole machinery of type 2 secretion system, which help the bacteria to inject this effector into the host cell, is dismantled. So that's what we hypothesized, that if we dismantle this machinery, the bacteria will definitely reduce its pathogenicity. So this was first target, but we learned after doing that it does reduce its pathogenicity, but it does not completely reduce its pathogenicity. Then we thought more deeper into it, and we decided to delete PUR-B gene. This purine synthesis process in bacteria is de novo process. It synthesizes its own AMPs, GMPs, CMPs. So these nucleotides are synthesized by bacteria by itself for its energy metabolism and for its DNA synthesis. So we found an enzyme which is very crucial towards adenyl succinate lyase enzyme, which is important for synthesis of AMP, adenosine monophosphate. So if bacteria will not have its own AMP, it will pick it from environment. And when it will pick it from environment, its growth will definitely be slowed down. That's what we usually want in a vaccine, that it should be growing less speedily than its wild type, so it can be managed by the immune system in that period of time when it has got the infection over there. So that was the idea behind this study, that we want to make it a slow growing organism with reduced ability to attack cells. So the first was knocking out of these two genes. So it took a long time for us to establish this technology in Pakistan. I did my PhD from New Zealand, and we were capable of doing this thing quite frequently in our labs. we were able to generate, in my study, 16 different mutants in less than a year time. But here it took me 7 years to create these 2 deletions. And it was resource intensive, and I was unlucky to get any funding. So it costed a lot of fortune of my own self. So it was, but my passion always remained live, and I followed this passion and we did it. So what we did, this is a pretty more deep science, but if you like, what we do that we replace the gene with an antibiotic cassette. The particular gene is replaced with an antibiotic cassette, and then we remove that antibiotic cassette to leave a scar in that particular gene. This type of gene, when we believe this type of gene is called isogenic knockout mutant. The remaining whole organism remains same, isogenic, and the only one gene which you are targeting is deleted or replaced. So we replace it with an antibiotic cassette, and then we remove the antibiotic cassettes. So how did we know that we are successful? If you look at there is 1421 base pair wild type gene. When this gene is replaced with a 900 cassette of antibiotic and some remnant genes, it becomes 1, 2, 2, 2, 1, 2, 2, 2, 12, 22 base pair size. That was the first proof that the gene has been removed from that location. Then another gene was SSAU, that was 1059 base pair, and the cassette we used, kanamycin, was larger. So when we inserted the cassette, we can see that the mutants got that same area using the same primers. We got a 1680 base pair DNA over there, which means that it has been replaced. And if you look at that, so if you look on the first 3 lanes, 1, 2, 3, in 3 it's a wild type, 2 when cassette was inserted, and 1 when cassette was removed and scar is there. Now this shows that mutant has been developed. Similarly, the wild type is 1059. When we inserted cassette, it's become 1680 base pair, and when the cassette was removed, it is 260 base pair remnant gene. This is called scar. So this was a genotype information that we were able to create that mutations. Phenotypically was very interesting, as it was expected that when you don't have the purines available made by the bacteria itself, the bacteria will definitely have effect on its growth. So we saw that wild type grows, you can see the square boxes, and the mutant was growing very slow. So to understand that whether it is really the adenine which is required for the growth, we used minimal media which only provides the known ingredients. So you can see that triangles, when wild type was growing, the triangles when wild type was growing, and then you can see squares that this bacteria was not growing. When we added adenine to the media, that started growing, which is compensation of adenine, which proves that it's the only adenine deficiency which is hindering that bacteria growth. Otherwise, the isogenic mutant was normal, which is a very strong empirical data to prove our hypothesis over here. So now that mutant was ready, now the next question was that mutant is not an answer to the question. The question was whether the mutant is safe for birds, still can be used in as safety, and whether it is a vaccine or not. So we did a lot of studies. If you look into the story that we inoculated our birds with wild type with the 10 to the power 5 to 10 to the power 9, and we see that mortality was 20% with 5, 10 to the 6, 30%, and 50%. And we calculated that Salmonella Gallinarum wild type, without any changes, causes 50% mortality when it is given at 2 into 10 to the power 8 CFU per ml. So that was LD50 of this organism we calculated, which we have got with ourselves. Now we compared it with so we use the same dose, 2 into 10 to the power 8 dose of the wild type and of the mutant. So if wild type is pathogenic and mutant has lost its pathogenicity, the hypothesis was that it should not cause any damage to the bird. So we measured different parameters. We measured body weight, mortality pattern, clinical symptoms, and gross lesion scoring. and persistent bacteria in liver and spleen. So you can see over here that first the body weight, generally the control which was not infected and the mutant used, the body weight remained same. So the bacteria did not damage anything, so body weight wasn't lost. But with SG wild type, the surviving animals lost their weight. But if you calculate in terms of mortality, the weight loss would be very prominent. And here you can see that the mortality in birds which were inoculated with the wild type went up to 80% till day 22. And with our strain, which is a mutant strain, the mortality was 0 till 22 we measured, and later in other studies, till all the life, there was no mortality. We also calculated the lesion scoring, how much pathogenicity this bacteria has caused, that what kind of pathogenicity it has got. Interestingly, if you look into the study, we did this by mathematical scoring, like we scored it if there was no clinical signs, if there was normal and active, we scored it 0, slight depression 1, moderate depression with drowsiness 2 score, severe depression with no responsiveness, we gave them 3 scores. Similarly, when we opened the birds, when we saw the diarrhea condition, normal color and shape 0 score, perianal contamination with feces 1 score, white viscous feces 2 scores, and green pasty vent is 3 score, which is very serious disease in case of Gallinarum. So you can see that our mutant, the double mutant, showed a very almost 0 score, meaning no clinical signs. And the wild type showed 2.6 or 2.8 score, which means that highest score almost. That's very pretty much for those who know that Gallinarum caused serious illness in birds. So all those signs were declared, which again proves that the mutant has lost its ability to cause disease. Thirdly, we did it spleen and liver scoring, and same thing we did, number of foci present on the liver and spleen, and it was very interesting to see that mutant behaved very extraordinary. And similarly, spleen's result was good. Then we also studied that how long it takes to get rid of the organs. So we learned that our mutant was capable of becoming 0 at day 21 post-infection or post-vaccination, whereas the wild type continues to stay in the host. So this is another very important feature which we believe vaccine should have, that live vaccine should produce titers, antibodies, cells, immunity, and should leave the host. Now, that was interesting. So we were able to prove that the vaccine is safe. Now, whether it is immunogenic, whether it provides immunity, so that was interesting. And we compared it with the only available vaccine in live vaccines, that is SV9R. So we compared and SV9R is quite good vaccine, I believe. And this vaccine which we produced was compared with 9R through humoral immunity. And if you look at that, its humoral immunity is almost same as 9R. Cellular immunity somehow behaved a little bit better than 9R. And if you look into the different parameters of health, that how these two vaccines affect the health of bird, so we were capable of showing that our vaccine is mild on bird when compared to the SG9R, which is like, if you look into that study, you can see that out of 20 birds, 1 bird showed mortality with this vaccine. In our case, no mortality at all. And control showed 75% mortality, which indicates that we were able to add some more value to this vaccine, the vaccine available, by making it mild and more protective, or equivalent protective at least. So this gives you, for example, if the mortality is 5% and you have 100,000 birds, so it means that you are expecting a 5% mortality, which is lost. that loss we can protect for vaccine because we believe that causes zero mortality and zero clinical signs. You can see over here that liver necrotic foci were little bit more than some numbers in case of SD9R, but in case of our vaccine, there was no foci at all. And spleen enlargement, spleen necrotic foci, again, our vaccines behave better than the only established vaccine in the world. And so this shows that our vaccine is very good at challenge and also very good when compared with the competitive vaccines. And so what we learned from this study and this, that whenever you attack your birds with oral infection, our vaccine provides very good immunity. But there was one still question. The SD9R is always injected in birds. Similarly, we were also not able to make an oral vaccine. Our vaccine was also injectable vaccine. The reason is that this salmonella is such a nasty organism that it survives and goes through the intestine, and this time it gives somehow some damage to the bacteria. So all vaccines in the world for Salmonella Gallinarum till now are injectable vaccines. So our vaccine was injectable with marvelous results you have seen. So we got a patent out of this, which is about to receive. We have got that patent, and this publication is the first publication with single mutation, and we are publishing this. This data which I have shown, I have not been published. We just published with a single mutant with Poultry, and this was our conclusion till for this study. But we left with one question. We were still enthusiastic. Is there any way that an oral vaccine can be generated? The rule is that you have an oral vaccine, you put it in the gel, you leave it everywhere in the country, all birds should pick it, and they should vaccinate. Then, so this can be the way to eradicate your Salmonella Gallinarum from your country. You cannot inject every bird in the field. You cannot inject every bird in the wildlife. So that was the idea, and no one in the world yet has submitted a vaccine which can be given through oral route to Salmonella Gallinarum. When we were talking about it, a new technology emerged, which is called gamma irradiation. If you cause gamma irradiation to the bacteria, bacteria's DNA is damaged, so it loses its reproduction, but still sustains its metabolism for a while. This is called residual metabolism. So that emerged as a very interesting idea, and we thought that let's try with our Salmonella Gallinarum. And very interestingly, when we use that technology, I'm not showing that how humoral immunity works, how safety works, everything was wonderful. I have not added that data. But in addition, what we saw is very interesting. Number one, it can be given through oral route. And with oral route, very interestingly, this is the first vaccine in Salmonella Gallinarum which goes through oral route. Once it goes through oral route, those who know immunology, they know that anything which goes through the natural routes, it goes through the epithelial cells. When epithelial cells will take an organism, live organism, they will present it with MSC1. When they will be presented with MSC1, then the only case CD8 T lymphocytes will be able to develop their cells. So they will be converted into CTLs. So this is very important phenomenon. That's what we see in live anti-vaccine and killed anti-vaccine. So the only live vaccines which can go through the epithelial cells can elicit CD8 cells. So that was our question, whether this elicits CD8 cells or not. And interestingly, when we did this study on flow cytometry, and this is the basic study we did with the control at SD9R, and we can see that the CD8 cells are generated, CD4 cells were counted, not counted, very well counted through flow cytometry. And if you look over here, so you can see that as compared to control, there was high number of CD8 cells and CD4 cells, which is interesting. And when we compared it with the SD9R, you can see gamma SG, this was the gamma-irradiated Salmonella Gallinarum, showed a very high quantity of CD8 cells, which gives a very promising result that this vaccine does not cause a single clinical sign and still taken up by the epithelial cells and enlist the CD8 cell, which we can see as the vaccine scientist was a dream that we can generate a vaccine, and we have been able to develop first oral vaccine of Salmonella Gallinarum, which shows robust cellular immune response. Thank you very much.